So our first speaker of this session is Martin Depken from Delft University of Technology, who will present his work on using mechanistic modelling to improve off-target activity predictions and elucidate the physical basis of Streptococcus pyogenes Cas9 fidelity. And obviously this is very relevant today, as obviously the Nobel Prize in Chemistry has been awarded for gene editing using CRISPR-Cas9. So over to you, Martin, if you'd like to share your screen.
Okay, so good morning, everyone. Today I'm going to talk to you about the work that we have done trying to understand the targeting behavior of Cas9, really from a mechanistic point of view. And the work is done by Misha Klein and Bruce Islami, and I am an associate professor at the Bionanoscience department in Delft. And given today, then I couldn't really resist putting in this slide here. And how suiting it is then that I also get the prize for the sharpest tool in gene engineering, and how suiting is it that I'm going to talk today then about exactly how sharp we think it is, or how we can understand how sharp it is. But before I get into that, I would just want to give a very, very brief introduction for those who have missed what this whole thing is. So the CRISPR-Cas system is an adaptive immune system that exists in archaea and bacteria. And so these organisms have a genetic loci from which they can express, among other things, the Cas9 molecule, and also transcribe, store viral sequences from past infections. So if they then have a repeat infection, they can load this guide RNA into the Cas9 molecule and then use that to attack then a repeat infection to the DNA and cleave that DNA. So obviously this is then what the whole excitement is about with CRISPR, because of Cas9 then that you can very easily load in and program it to hit different positions in the genome. Now, of course, that sounds like a very good scalpel then, that you go in and genetically do engineering. But there's also been a lot of studies reporting off-target effects. So there is an ongoing debate on how precise this tool actually is. And here's just a few studies come up then, discussing the off-targeting in this system. Now, of course, it's extremely important to try to characterize these molecules experimentally. But if we want to be able to characterize all potentially interesting guide and target combinations, it would be in the order of trillions of different experiments we had to do. That's obviously not possible. So therefore, there's a lot of effort in the field now to also do a computational approach to try to actually predict off targets. So to date, the most of these approaches, or almost all of them, are top-down approaches, where you feed in a genome and a gene to your computational pipeline. And then on the other side, you get out a ranking, where these are then ranked according to idiosyncratic measures of each of these methods. And so you get a whole genome, and it is ranked, all the sites. And then you introduce some cutoff somewhere, and then you classify the targets above that cutoff as strong off targets, right? But this is a very non-quantitative approach, and we know from experiments, so these are human cell lines, that really the activity on off targets, so you can see here, so this is a dosage response of Cas9 for various off targets. Here is the on target. And you can see that it depends on the concentration, which is quite natural. But that should then give pause for these methods that don't take anything like concentration or exposure time at all into account. So really the problem, or the question I then asked Misha and Bruce, what if they could take what we know molecularly, or the molecular knowledge of this system, and kind of lift it up to a level where we can quantitatively predict whole genome activity, not just score off targets, but actually predict the genome activity in a way that would adapt to concentrations or exposure times, right, like this. So, of course, since we want to predict it over a whole genome, we will need some high throughput experimental input. And on top of that, not so much next generation biophysics maybe, but old school statistical physics will help us in this process, right? Because we really know quite a lot from statistical physics on what to expect of these small systems. So the system, the basic reaction we want to describe is this one, right? So you have Cas9, you load your gRNA, and then you complement with the target strand, and eventually cleave. Turns out that this middle part is quite complicated. So this is a recent paper where they used a magnetic tweezer assay where they have a rotor bead. And then when you have hybridization between the gRNA and the DNA, you start unwinding the R loop, and then you will actually see the bead turning in this assay. And they can then track how long the hybrid length is here. And you see that it actually varies quite a lot, and it can be many positions. This hybrid length can be in many positions then. So we need to have quite a detailed model to capture this. So what we start with is just this. So the states that we have is that you can either be in solution, then you can bind, and this should be saying that all this is kind of understood from structural data quite early on. And then once you've bound this PAM, 3-nucleotide PAM sequence, it's a motif on the protein that recognizes 3 nucleotides. And then you can start then opening up this, or making the hybrid opening up the R loop like this. But of course, if there's an off target there, maybe you will reverse and then unbind again. But if it's a good target, maybe we'll go back and forth a bit, but eventually then reach the full hybridization and then cleavage ensures. Right, so the model we have is simply this, right? So you have a unbound state, and then you bind the PAM, and then you have a lot of other PAM states or other hybrid length states in between, and then you have a final cleaved state. The good news is that this is a solvable system, so we can calculate everything that we want, all the physical quantities we want out of this. The bad news is that to fully determine this, then over all possible guide and target combinations, you would need something like 10 to the 26 rates to do this. And it's obviously quite useless if you try to kind of compare it to data or something. So we have to come up, or Misha and Barutz had to come up with some simplifying rules. And this is then our mechanistic modeling rules. So we make 3 basic assumptions. If you want the details, you can find it in the preprint that is down on the screen there. But the first assumption we make is that the mismatch type has little influence, right? So meaning that we only take care of that if it's a mismatch or not. We only keep track of that. The other thing is that we say that the energetics of this complex is local. So the energy cost of having 2 mismatches in should be the same as the sum of the energy cost of each individual mismatch. And then on top of that, a lot of the experiments are performed on a version of Cas9 that is deactivated. So this is called dead Cas9. And our assumption is that we can translate between Cas9 and the dead Cas9 just by assuming that everything is exactly the same, apart from that the dead Cas9 hasn't got the ability to cleave. So it lacks the bite. Right? So with these 3 assumptions, we can actually bring down the 10 to the 26 parameters to 46 physical parameters. And it remains to see if any of the physics is actually left in there, right? So this is quite a drastic reduction, but 46 parameters is still quite a lot. So we teamed up with the people from Ilya Finkelstein's lab. And what they have is these really fantastic experiments, these next generation sequencing experiments, where they can, for a very large library of guide and off targets, they can determine the cutting frequency or the cutting rate. And they can also determine the association constant. So they do this by doing the association constant, they do sweeps. And so they get these 2 numbers out. So it's a kind of low resolution experiments in the sense that we only pick out 2 numbers from each, one number from each experiment, one from cleaving and one from binding. But then there are many of them, and we combine them. And then the hope is that this still should help us determine what goes into the model. So what we use for training here, so we now want to determine our 46 parameters, is the set, while it's on target, the set of all singly mismatched on target, like I'm trying to show here, and the set of all doubly mismatched targets. And then we just turn the physics. So if you have a parameter set, we can then calculate exactly what they get in the experiments. We can calculate the same quantities. We can do the same fitting to pull out the same numbers. And then we just directly compare this to the experiment. And we put it on a cluster, and we do a stochastic minimization of the least square deviation in this problem then to see if we can determine all the rates. So what I'm going to show you now is exactly this, right? So this is our training set that we've used. You're going to see a movie here of 30 of these optimization runs. And what you see in the top left here in black is the on-target landscape. So this is the free energy for different hybrid lengths form. So you see the hybrid length on the x-axis there and the free energy. And this is the initial condition we set. And also you have the mismatch penalties in red there. And on top of this, I'm always also going to show you how the fit is actually now being approached by the on this singly mismatched sets, as you see here. And so initially you see they have a lot of data points there and the fits don't look so good, obviously. One thing that should be highlighted is that these data points, they seem to have a lot of variation in them, but this is not noise, this is actual data. They are very reproducible experiments, this. So let's now run it and see if we can see some structure emerge here, right? So these are now the free energy landscapes. I hope it's not too slow over the web for you to see. But what you should see here is now as the fit starts becoming better and better, there is really something, a free energy landscape crystallizing out here now, right? So on the on-target landscape, we seem to see we have now, if you can see my pointer, we have a, 3 metastable states here, it seems. And then we have some transition regions here that are not very well determined by the model, but that's also kind of expected, because if you have a system controlled by these metastable states, there are many realizations of an intervening barrier that can give the same effective transition rates over. So the hope to actually determine these is not so high, but and then we also see that we have some definite structure here also in the mismatch penalties, but in general, it's around 5-6 kBT. All right, so now we can then see, first of all, of course, it looked like we can fit the training data quite well, and this is another representation of this. So these are for the binding experiments, and where this delta ABA is proportional to the log of the association constant. And we see for 0 to 2 mismatches, we do a very, very good job at predicting what they would get in the experiments, but that's also expected since this was a training set. What was not in the training set was for all the other higher mismatch things. And we do as well actually predicting those. So that's very, very encouraging for us, because that was not in the training data. On top of that, we can also predict completely different experiments done by a different group. So this is from the Greenleaf lab, where they measured the effective on-target rate. And this is a prediction from our model that is parameterized on the other data set then, but in no extra fit parameters here. So we can just predict this straight off, which works pretty well, right? All right, so our goal was really to try to understand how this targeting behavior works. And maybe we can now see if we can connect it, though, to the single molecule experiments that have recently been performed. So these are the single molecule experiments I was talking about before from Ivanov. And you can see here, right? So this is the on-target landscape. So this is binding on the on-target. And you start out in solution, obviously, then you don't have any hybrid unwound. Then you find an intermediate state here, which we also identify on the right place. And then you end up in the cleavage competent state in the end. Now, if you put a lot of mismatches in the beginning, so you can see the sequence up here, so you have excess or mismatches. If you put in a lot of mismatches in the beginning here, of course, then we abort all the binding whatsoever. And that's also seen in experiments. On top of that, we can put mismatches in the end. And compared to what you see single molecule experiment, we put mismatches back here in the end. You get a wall in the end, and not surprisingly then, when you do your binding, you bind in, but you just bind into this intermediate state in the beginning or in the middle there. Maybe more interesting is if we put a single mismatch at position 18 here, so that's in here, and then you're, even though it lies in this forest here of undetermined free energy values, you can see that it has risen the free energy values at the end here of all these. And I forgot to say, but all these solutions are essentially as good, right? So these are 30 runs, and they're all within 15% of each other in minimizing the least square. But what you see here is that you get the region in the minimum region seems to be somewhat enlarged, and that you can also see in the single molecule experiments, where you see now you start transitioning between many, many more discrete states inside this region. So this we were very, very happy with here now that we managed to recreate the single molecule experiments, and we really think that we understand how this system works on any guide target combination. But we wanted to compare it to something else, right? Something more quantitative. And we thought we would wanted to compare it to the off-target predictors that are out there. So, but that raises a question. So we actually calculate the activity over a whole genome as a function of time and concentration. So we can get an analog signal here. whilst others, they score strong off targets. So this is more of a digital signal. So how do we compare our two results here, right? So what we are forced to do a comparison is actually we have to do quite a lot of violence on our own model and try to reduce it into a simple on-off target predictor and classifier of that. And we do that by just ranking all our sites based on the cleavage, the relative cleavage rate in the low concentration limit. That's a choice that we made. It seemed reasonable, but you know, you could come up with many other choices possibly. So by doing that then, right, so this is what we do. We have to reduce our little model into something a bit smaller. And what we have, the result of this is going to be graphs like this. And I just really, these graphs show the results as you sweep this threshold for what you classify as a strong or a weak off target. And it keeps tracks then how the precision, you know, so the fraction of predicted cleaved sites that are actually correct, and the recall, the fraction of cleaved sites identified by the model. So it keeps track of these two numbers as a function of the cutoff, and as you sweep the cutoff, you get these lines. Thing is that you want to be out here, you want to catch every off target, and you don't want to catch anything but the off target, right? So you want to be up there. So you can see none of the models are competitors. So these are the two strongest competitors we have. We are actually, there is one more that we're in the process of actually adding to this graph, but I'm pretty sure that we're going to beat that one too. Anyway, so you want to be up there and this is then for the EMX1 gene. We can do this on many other genes to compare over the whole of the human genome then, and we'll compare to other models. And this max F1 score is a summary of the graphs like this. It essentially tells you how far from this point you are. And if you look at all these scores, we also, on all these other genes, we also beat the competition. And this is kind of interesting in the sense that we are not including sequence at all here, right? But all the other models are including the sequence, and we still beat the competition. The one point that we don't seem to beat the competition over here, but that is also due to that this actually formed part of the training set for this model. So it's not so surprising that they do quite well there compared to also too, right? But still we do as well. So this was, this we're very happy about this, right? And this is extremely exciting for us. So now going forward then, we would want to, you know, so of course the interesting thing here would now be so we have this way of classifying these molecules. We can pull out their free energy landscapes from the high, you know, from these high throughput experiments. So what we would want to do now is really to see if we have these high fidelity mutants. Maybe we can pull those through a pipeline and actually see what makes them high fidelity. Is it that they destabilize the PAM distal part of the matching here or not? Well, but for this, we have the data, but we haven't actually been able to run the things yet. So this is to be continued. And with that, I would like to thank you all for your attention and thank for all the people that were involved and the funding. And over to you.
Thanks, Martin. That was really interesting. Chris, have we got any questions?
Yes, we have one question. Well, actually, two questions from one of the attendees. So the question is whether you can do better by relaxing the assumptions that you're using. It looked as if you're doing pretty well to me, but the question is, can you do even better?
Yeah, I mean, the hope is that, obviously. So we focused first, we thought it was, you know, we get the physics right. And then we start adding all these details like the sequence and all this. And this is certainly things that we want to do going forward. Of course, those problems turn a lot harder because now you have a lot more free parameters to fit, right? But that's certainly things that we're going to do in the future.
Okay, and the second part of the question is whether you can use some sort of maximum likelihood approach for inferring the parameters directly from the data.
Yeah, so now we just do the least square minimization here. I don't really think that the maximum likelihood approach will probably be applicable to these type of experiments, but if we do the single molecule, if we can compare to single molecule experiments, I think that's where these are going to really enter, because there you get distributions of off times, for example, and then you can really easily treat with a maximum likelihood approach. So one thing that I want to mention with this is that, you know, since we have a physical model and we can then also, so we have a core model that we can then couple to whatever experiment we want, right? So in principle, we could also use, we can train it on these high throughput experiments and add in some single molecule data at the same time if we want it, right? And that's where I think that the maximum likelihood approach would actually be the best suited.
Okay.
So I think David had a question.
Yes, David.
Thank you, Geoff. Very nice talk, Martin. I really enjoyed the talk. I was curious about your assumptions about the nature of the mismatch and the additivity of the mismatch in a way. So when the mismatches become really close, you're going to start breaking down the nearest neighbor's rules for the DNA stability. And also, you know, pyrimidine versus purine mismatches might have very different results in the R loop. What do you think about that?
Absolutely, right. So this is even more violence on the system than the nearest neighbor model, right? Because this doesn't even take into account any neighbor. So even if they sit right next to each other, they don't feel each other. These are definite ways of improving this too. We could think about using the parameters for the nearest neighbor model that is already parameterized to lower the number of fit parameters that we actually need to include. So that's one thing. Looking at different bases, not looking at all four, you know, splitting them up. Yeah, there are many of these things. These are all refinements that we are definitely looking into doing going forward. And another thing too, right? So we are now coupling. I mean, I know you are interested in the force behavior of this system. So we are now adding models where we, you know, torque to the system, so we can actually model that too, and force is also in the works.
Excellent, thank you.
Cheers.
Okay, we have one more question if we have time, and that is whether you have plans to run these models for other Cas9 variants.
Oh yes.
Then a very common question from experimentalists is how they would use the model, your model, sorry, to inform protein engineering approaches. Yeah,
so this is something that I would really hope that the model will be used for, right? Because, you know, if we do, if you have these free energy landscapes that we showed you, right? And the hope then is, well, that they will come, where did you have them? Here. So if you have these free energy landscapes, of course, that, you know, how they are changed depending in these mutants. And we actually have the data from ILIA already with this, right? So this is something we're really excited to start doing now. Yeah, so I don't know what will actually happen, but one thing is that I think would probably, many of these high fidelity mutants are probably is that they have destabilized the PAM distal part here. So you would see that the free energy barrier here would rise up, right? So how that would be used in protein engineering, I don't know, but I would hope that they could use this type of analysis and characterization of these molecules to get a better insight into their molecules, the engineered molecules.
Perfect. So I think we're just about on time. So thanks again, Martin.
Thank you all.